isn't met in those other areas anymore, it just kind of confuses.
Okay, so I think you're getting at what I'm thinking of here. Transcription factors expressed in discrete areas. And, sorry?
Well, it probably regulates them in other areas.
Right,
As the gap sheets do, so then it's making other areas not have anything. It's increasing enough. Where it's increasing, it's making places around that decrease because it's regulated.
right, so we have other regulators in the milieu here. And in fact, what's happening is it's being regulated perhaps by big-voided nanos, by some of the gap genes, but it takes time for the parallel genes to get turned on. At first, they're not there. Then once we get to this stage in embryo development, they turn on. And they themselves are also another regulator of each other within the class. So initially, we might have some input from the gap genes and the maternal products about where FTSE-TRASA can be expressed. But then once the parallel gene transcription factors are made, they add in. And so collectively, we're getting more and more regulators contributing and more crispy, refined expression for this particular gene product. So point being that the parallel genes themselves also contribute and help sharpen the domains of expression for sibling parallel genes. Okay, so we have then parallel genes expressed in these stripes, and there's one more thing I needed to tell you, which is how is it that we can actually arrange for a parallel gene to become expressed in a stripe? Now, in the case of the gap genes, we said it's responding to different transcription factors that are present within that territory of the embryo. And in essence, that's going to be the same paradigm for parallel, but there's a quirk in that, stripe number 1 sees a totally different combination of transcription factors than stripe number 8. Just bigoid is going to be quite different, for example. So, how does the embryo accommodate this so that it can get even skip2, a parallel gene expressed in the correct location? So, to look at this, we considered the enhancer regions for this particular gene, even skip2, and we'll consider what it would see in stripe number 2. So stripe number 2 is fairly close to the anterior, and we're going to get fairly high levels of bicoid. We're going to get fairly high levels of hunchback. Giant is one of the gap genes, and that seems to be expressing at high levels, more anterior to this, but dropping off right at this point. Cruple is expressed in the posterior, and it's also just starting up right at this point. So for the even skip2 gene, it has a very complicated set of enhancers and promoters. And it was found that you can actually define a discrete region within those enhancers that respond to the situation for stripe 2. And so if you map this out here, here's our chromosome, and in kind of gray blue there it says coding. That means the coding sequence where the open reading frame is, and you would see amino acids being put into a transcript here. Well, you know what I mean. This is the coding region where the open reading frame is. And then all these other colored bars, these are the enhancer regions where you have the DNA that is responding to the transcription factors present in different stripe territories within the embryo. And you can do these little experiments where we snip out the region for stripe 1 and stripe 5, and then clone that in front of some kind of reporter gene, like GFP. And based on that, you would really predict that now you're going to see GFP expressed in stripes, but because we've only added the stripe 1 and stripe 5 enhancers, we should see GFP expressed in the same territory as stripe number 1 and stripe number 5. Because this is what you need to produce expression for the gene in that particular set of cells. So we can clone it in front of, we could use LAC-Z if we didn't like GFP. And then what you should see is a particular response. So here's an example of what that looks like. So they've done a double staining here where the light brown is just every single parallel gene, and then the dark blue is the LAC-Z reporter gene from our transgenic situation where we have LAC-Z with strike 1, sure enough. So these DNA sequences and then strike 1 enhancer can promote LAC-Z expression. We could do it for strike 5. Shows you it's a totally different discrete territory or regulatory region that governs expression in strike 5. Or we could mix and match them together. We could also take this same construct and put it in a mutant background. Okay. So here's a case where we're missing the wild type contribution of giant, which is one of the regulators of expression in the posterior domains. And in giant deficient embryos, now we've lost one of the constraints for expression in that territory, and we've seen an expansion of the strike 5 to quite a wide width. So basically, the same sort of principles are applying, but what we have to accommodate the striped nature of expression is a unique and discrete region within the DNA regulatory sequence to take care of each particular stripe. So, you can imagine this is kind of like a big territory for the upstream and downstream regions that is needed to get appropriate expression for these coding products. Okay, so what about within these discrete DNA areas, within these enhancer modules, what do we actually see? So what is the nitty-gritty of what it looks like inside the enhancer that's going to promote expression in this narrow group of cells? And here's a figure from your book. It's kind of complicated looking, but each one of those little colored bars is now the specific and small recognition site for each transcription factor. So you could recognize a lot of B's in there. Those are bicoid binding sites. They may only be 4 or 5 base pairs wide, but that's what bicoid recognizes when it's scanning around promoters and enhancers, and if it finds it, it'll find that place. So many of these within this, 25 different transcription factor binding sites, some of those are sites that would stimulate transcription if the transcription factor is present. On the other hand, some of them are sites that would tend to hold back or repress transcription if that factor is present. And according to this principle of combinatorial control that we talked about in one of our first lectures, we have that scenario. We have both enhancers and repressors all kind of in there tweaking the thing so it's going to be expressed at exactly the right level, the right time. Remember, timing is important in development, and the right space. So the amount, the time, and the space are all features of to what degree this gene is quote-unquote turned on. Okay. Now I think we're ready for segment polarity genes, which is another class that's turned on a little bit later than the parallel genes are. And you can see the embryo by this point has finished up with the cellularization process and is beginning to undergo gastrulation. So that's why they have a little curved and indentated shape there, so that's an embryo undergoing gastrulation. And if you would look by in situ hybridization or immunohistochemistry at some of these segment polarity genes, again you get a striping pattern, but this time we're going to get 14 stripes instead of 7 stripes. And those stripes are going to be narrower than the parallel stripes. So they may only be 1 or 2 cells wide. These are also transcription factors. 1 or 2 of these in this class are paracrine factors. That's our only exception here, is that within the segment polarity, some of these genes are secreted short-range signaling molecules, but everybody else on that whole slide there is transcription factor. So now we have to explain how we're going to get expression within these 14 stripes, and what does it mean that they're expressed in these territories. So here again is showing you segment and parasegment and how they overlap by 50%, they're offset 50%. And in the larvae, you can recognize within one segment here, about half of it has bristles and half of it does not. So there's really a distinct sort of anterior half, posterior half feature to this. And you might expect then that there are different genes that might be expressed in anterior halves or posterior halves of segments as time goes by. And this is the case where the segment polarity genes is now, instead of being expressed in every other parasegment, they're expressed in every single parasegment, but only half of it. So here's our little thing with the 14 segments, and you can see then 14 stripes accommodating expression in only half of each one of those segments on the parasegments. And then, just like we've been talking about before in principle, when you mutate one of the segment polarity genes, you're going to get a smaller larvae on the anterior posterior axes, but the patterning of that larvae compared to a gap gene or a parallel gene mutate will be a different phenotype. This time we're missing a portion of every parasegment, aka a portion of every segment. Okay. What is next here? Okay, the last class of genes I'm going to talk about before the break is this group here called homeotic genes. And they are expressed in the embryo somewhere around the syncytial last germ stage or later. And they're expressed in unique territories, but it's not so much a striped pattern and not so much a, well, I guess they're kind of broad bands of expression. But the thing about this group of genes, and again, we're talking transcription factors here, is that they have some incredibly interesting mutant phenotypes. So these genes were found in Drosophila, and in fact, when we look, they all map to the same chromosome. So it's chromosome number 3, and it contains 2 clusters of genes that add up to 8 genes altogether. So there's one cluster here known as the Antennapedia complex, a group of 5 genes, and then slightly downstream from that, a second group called the Bithorax complex that contains 3 genes. And these are actually responsible for specification of body parts. That is, that it's going to help assign the identity to that body part. So cells in there will know, should we become thorax that has wings and legs? Should we become abdomen that has bristles of a certain type? What should we do? And if you mutate any of these genes, you end up getting a fairly drastic phenotype because the identity of those cell types has been either altered or wiped out. So another thing to point out here is we have a certain order. And you don't need to know the gene names, but let's look at red, yellow, purple, orange, and pink. And then when you look in the fly, the order of the genes on the chromosome matches the order of the genes are expressed in the embryo. So this one here, the red one, is on one end, and that's expressed in the head. Then we get yellow, that's expressed in the neck region, if you will, or the first thoracic segment. Purple for thoracic segment 2, and it goes on down the line. So we're getting this correspondence between spatial expression in the embryo and order of gene on the chromosome. And that scenario is called collinearity, which I think may be written on one of the slides down the line here, but it's a very rare condition. In fact, this is the only situation that I know of where we have this correspondence between expression pattern and order of genes on the chromosome. Normally, you can get any number of genes on a chromosome. They have absolutely nothing to do with each other in terms of function, and there's absolutely no correlation about when or where or whether those genes would be expressed in the embryo in any kind of pattern together. But for this group of genes, there's this special sort of cooperative or at least ordered type of expression within the fly. Okay, any questions on this? Okay, this lecture is going to be on your following exam, not the one we're having on Monday. So let's go ahead and take a break. We're at 8:48, so let's come back at 9:48 to restart.